Okay, Sandra, do you want to take it away?
Yep. Hi, everyone. Welcome to the Black in Comp Bio workshop series hosted by the Black Women in Computational Biology Network, or BWCB. BWCB is a 501c3 nonprofit organization whose mission is to accelerate opportunity at the intersection of biology, math, computer science, and computer science for Black women globally. We have about 300 members around the world, and we have over 3,000 supporters in the network. My name is Sandra, and today we present the Black in Comp Bio workshop featuring Dr. Jasmine Baker. After this workshop, BWCB members do have an opportunity to meet with Dr. Baker for 30 minutes in a separate Zoom. So please let us know so that we can send you the link to the Zoom for that meeting afterwards. To introduce Dr. Baker, she is a passionate bioinformatician with a PhD and over 10 years of experience in scientific research. She excels translating complex scientific concepts into clear and engaging communication for diverse audiences. Dr. Baker leverages her strong foundation in life science techniques and data analysis to spearhead community outreach and engagement for the Open Cravat software. In her previous role, Dr. Baker thrived in a collaborative research environment, instructing and training personnel, developing protocols, and contributing to peer-reviewed publications. Her problem-solving skills and enthusiasm for helping others translate seamlessly into the exceptional technical support within the Open Cravat community. Dr. Baker possesses a comprehensive bioinformatics skill set encompassing programming languages, machine learning algorithms, and data visualization tools. She is also a proficient in wet bench techniques and various software packages commonly used in bioinformatics workflows. Dr. Baker's dedication to clear communication and fostering a thriving Open Cravat community makes her an asset in advancing open source scientific software. Please join me in welcoming Dr. Jasmine Baker, who will present today, Get Started with Open Cravat, a tutorial for annotating genomic variants and hands-on demonstrations and real data. Dr. Baker.
Good morning, everyone. I want to welcome you to the webinar. I'm very excited to have everyone here today. So I am the lead of outreach and training with the Karchin Lab at Johns Hopkins. They focus on cancer variants, and I'm going to be discussing their awesome open source software entitled Open Cravat. We also have Dr. Karchin, who's on the Zoom today, and she will be manning the chat box along with other members of the Black Women in Computational Biology Network. So if you have questions, please feel free to put them in the chat box and either Rachel or someone else will respond. Hopefully everyone was able to utilize the pre-webinar checklist that we posted on social media to install Open Cravat, either through the terminal to do a local installation, or if you use one of the Windows or Mac installers, you should have been able to do that as well. If not, there are other options for you to use our hosting webpage. So if you just want to follow along with one of the examples, and you also have the option to just follow along today visually, which is also okay. All of this information will be recorded and shared later for you to view and follow along with. We will also have slides and things available via our discussion board and our landing page. On the slide that we have here, I have a QR code that leads you to our discussion board. And on our discussion board, this is issue number 271, and it gives you all of the instructions on how to download Open Cravat, where to find the zipped data files that we're going to use today for our examples, and a list of the annotators that you will need to have installed with Open Cravat. If you were not able to install all the annotators by any chance because you didn't have space or you just didn't get a chance, that's okay. If you just let us know which ones you have, we can go through it offline or afterwards. Or if you have a question about what I'm seeing versus what you're seeing, you can just put it in the chat box and we can kind of walk through what the differences are. I really want today's webinar to be really a little interactive. So before we get started, if you were able to install Open Cravat, could you please use your reaction button to give me a thumbs up, thumbs down, some kind of reaction, anything. Are we getting some reactions?
Yeah, I think it peaked around 8,
Okay. Okay.
more than
Great.
8, maybe half people.
Okay. Great. I can't see them on my end in the presentation mode, so keep. All right. So we're going to get started. So let's get started with Open Cravat. We're going to go through some tutorials on how to annotate genomic variants, and we have some hands-on demonstrations with examples and data from literature that you can actually go back and read for yourself. This is published work, and we also have some real data and a 23andMe example. So before we begin, I want to take a moment to get some insights about each other. I know this is a short webinar and we won't have time to actually introduce ourselves, but I would love to get everyone to take a moment to scan this QR code or go to this link, and the link's going to be pasted in the chat box. And just give us a little information about your background, what type of variants you may have worked with before, haven't worked with before, just so we can get to know each other and I can know whom is here today. And if you guys have questions, I'll have an easier way to, connect with you and help you guys out, especially if you would like to use Open Cravat in the future for your research or projects or personal use. Okay. Mary, I am teaching using the local installation version, but the screenshots are very similar, so you should be able to follow along. And there are similar buttons on how to upload the files that you guys are going to use that were on the discussion board that you downloaded. So I think you'll be fine in regards to following along if you're attempting to use the web version. Okay. Hopefully everyone has been able to scan this QR code or go to the link. I am going to post the link for the instant insights into the chat box. So I don't see it there, so I'm going to do that. And we will be good to go. All right. And thank you all for taking the time to do that. So let's get into it. So we're going to introduce Open Cravat. We're going to go over some use cases and then go over some 23andMe examples. If we can annotate it, we can understand it. There is a vast amount of information from all of the big data and the genome studies that are going on. Since the sequencing of the human genome, there's been so much data that's out and available for scientists to make scientific and medical insights from. However, there is a lack of appropriate annotation and emphasis and insight that's actually being made from that information. And this is a famous quote from the Karchin Lab, and they are very adamant about being able to annotate things so everyone can understand it and be able to make better scientific and medical insights. So what's the importance of variant annotation in genomic analysis? So we have a few key points here that I have. It provides crucial insights for interpreting the genetic variants effectively. Annotation also helps guide medical decisions based on being able to identify genetic variants that one person may have in comparison to another, which kind of hits the tip of the iceberg in the personalized medicine school of thought that a lot of people are leaning towards these days. Also, being able to understand variant annotation it helps you enhance your research and discoveries. Once you have these annotations, you can contribute appropriately to the advancements in scientific literature, discoveries, and within precision medicine. So, as I mentioned previously, the Carchin lab is predominantly a cancer lab. So I'm going to give a brief introduction to the types of cancer variants. So we can just kind of have a basis and a foundation laid before we actually get into using the software. So there are two types of cancer variants, germline and somatic. Today we are going to focus on somatic variants, but I'm going to give you guys definitions and explain the images on the screen and explain the difference between both. So germline variants are variants that are present in the gamete, and that variant is present in every cell in your body, including those in the germline, and the variants can be passed on to offspring. However, in contrast, if you have a somatic variant, it's completely absent in the gamete once the sperm and the egg create the zygote, and variation occurs during either the fetal development process or at any time after birth in any cell in the body except those of the germline. And the variants are not passed on to the offspring. So today we're going to focus on these somatic variants that typically that occur after the gamete is formed. And the variants that we're going to focus on are specifically from cancer tumors. So I have here the evolution of cancer in cells. So we begin with our normal cells, and there's a mutation which we have in our lighter blue field little cell. That begins to multiply. We're entering the space of the cell cycle that's being changed now, where we have rapid growth, and eventually you end up with a tumor. So cancer only develops when you have several mutations, which we see here, that accumulate in the DNA of an individual cell. So our samples are from a tumor, so we have an accumulation of these mutations. And then eventually at some point, as the cancer develops, you get a malignant tumor if the cancer is not stopped early enough. That malignant tumor, those mutations can be transferred through the bloodstream, and that's how we get to a metastatic tumor. So whenever you hear malignant and metastatic tumor, malignant, you can think of staying in place. And when you think of metastatic, that means the tumor and the cancer has spread to other organs besides the organ of origin. Specifically within those tumor cells, we are going to focus on cancer driver genes and mutations within those genes that are causing that are contributing to the oncogenesis. So there are two types of cancer driver genes. You have proto-oncogenes and tumor suppressor genes. So proto-oncogenes, which are here to the right, are before the mutation occurs, and tumor suppressor genes are genes that are used to, the name suggests, suppress tumors. So we are specifically going to look at, in our analysis today, we're looking for driver mutations. And when we say driver mutations, we're looking for mutations that are happening so early in the process, or that are happening that are actually driving the oncogenesis and the cancer to progress. So in the image we have here, we have our mutated tumor suppressor gene, and we also have a mutated proto-oncogene. And what happens is when you have driver mutations that happen in one of these types of genes, you have changes that are abnormal to the actual cell cycle. So you may have a loss of function, a gain of function, and that all contributes to these cancer cells developing. So now that we have our foundation laid for the types of cancer variants we're looking at, how cancer in cells evolves, and what driver mutations are, we're going to move into a little discussion about Open Cravat. So Open Cravat stands for Open Custom Ranked Analysis of Variants Toolkit. And just as the name suggests, it is really a toolkit. It's almost like you go into your garage, you open up your toolkit, and you have your hammer, your wrench, your screwdriver, your flashlight. You have so many tools at your fingertips. You will be able to do any job that you are aiming to do. So it is open source. It is housed on GitHub, which is amazing because the computing community is really big on open source. Open source allows for lots of users to be able to access it, and it's available to everyone globally. It's used for annotating, prioritizing, and analyzing variants, so germline and somatic. Even though we're focusing our most of our examples today on somatic, we will do a little. There are some examples of people using the software for germline that are in the literature. There is an annotation app store with visualization widgets, which are very helpful for visualizing the vast amount of information that the annotator helps you comb through. There are input format converters. So, for example, the 23andMe data that we're looking at today, that data has to be converted from the 23andMe format. And there are output format reporters as well. And Open Cravat is really great because you can utilize it to share and integrate different genetic annotation resources. So it's really great, especially if you're working on large collaborative projects, or if you have some data that's new and there was some data previously from years ago, you can still go back and use all that information to either share or integrate everything into one source. And we also have the citation at the bottom. So whenever everyone decides to use Open Cravat, here's the citation. So you can cite us. So the example that I have today is using the terminal. I chose to use the terminal for my installation. You may have decided to use the Windows or the Mac installer. And that's okay. That's okay. If you use the Windows or the Mac installer, you can just double click your executable app on your computer if you'd like to follow along. Hopefully you have downloaded the zip files from the discussion board. If you haven't, you still have time to do that, or if you prefer at this point just to kind of watch the YouTube video later, that's a-okay as well. So if you use the terminal, access your terminal, or access your app if you use the computer installer. Once you have Open Cravat running in your terminal, if that was your method of choice, you're going to type OC, which stands for Open Cravat, and GUI, graphical user interface, and you're going to have this nice wording of Open Cravat, and it's going to tell you that Open Cravat is served at whatever your local host is, and you should have another window that pops up, and it will take you to the Open Cravat GUI. Alternatively, if you did not download Open Cravat and you'd like to follow along with one of the files, you can use run.opencravat.org. However, I will note that if everyone decides to use that option, it might be a little lag real time. So just give it a moment and be patient. It's just like any other computer. You load it up with a lot of things at one time, and that's, sometimes things happen. So just be prepared if that does happen. Because when you're in real time, you just never know. So you can access the Open Cravat server here. You can log in or register yourself, or you can also try it as a guest. Both options are very functional. So next, we're going to move into what the actual workflow of Open Cravat is. So while you guys are loading up your terminal or downloading your files, if you haven't, or deciding that your method of choice to run Open Cravat, I'm going to go through a little bit of the workflow. So this is what the workflow of Open Cravat looks like. We start off with the input file. So your input file can be a comma separated value file, a DB SNP file, a VCF file, a 23andMe file, or an ancestry file. Today we're going to work with SQLite files. That's going to be the input that you guys have that was zipped. That SQLite file was it was created from VCF files that we had of data. Next, once you have your file input into Open Cravat, you've chosen your, you're going to go to the module store here and you choose the annotators. So once you choose the annotators that you would like to use, there are a variety of options, and I have a screenshot coming up to discuss that. You're going to move into actually using Open Cravat. You have a variety of external outputs that you can get. And then you can go through and analyze your results line by line and click through and kind of get into the scientific rabbit hole of data. So these are just our input formats again, and I just want to stress to you guys that normally we have to run the job, but we're giving you a pre-processed job today. So your input is not going to be a CSV, DBSTEP, VCF, Ancestry, or 23andMe file. Now I'm going to discuss the actual module store. So this is a screenshot of the module store. And if you have Open Cravat up and running, you'll see that you have these 3 tabs at the very top left corner. So you have jobs, store, and single variant. Jobs is where you would actually upload your input file, but we're going to talk about the store right now. So you have a really nice discovery button here to the left, and it gives you information about the most downloaded and new annotators that are available for you to use. A lot of these that you see on the screen now, like Civic, Gene Ontology, Form GKB, we're going to discuss those today. We also have the packages option in the store, which is here. So if you unclick Discover and you click Packages, we have a drug interaction package, rare coding, hereditary splicing package, and pathogenic variant package. All of these packages are great packages that are currently under development. So we have things within the packages. However, there are certain aspects that are still in development. So if you choose to use one of these packages and you notice something that maybe needs to be there or something you would like to contribute, because we are an open source software package, we are more than happy to have people contribute and develop their own annotators or packages if you'd like. This is also something very key to keep in mind when you're using Open Cravat. This option that says update available, it's typically good to pop into the store before you run your job, just to make sure that all of your annotators are up to date. Our software engineering team, they work very hard to make sure that the annotators are updated on a regular basis. So you may have already had a version installed and the next day there may be an update. So just pop in and click update available. And if there is an update available, you can press update and it'll update your annotator and you'll have the most recent information from Gene Ontology or whatever annotators you have installed. The next tab that I want to point out to you guys is the single variant tab. And once we go over this tab, we're going to pop into actually looking at some data. So this single variant tab is really great for researchers or people who are just starting out with their project and they say, hey, I have this one variant. I'm really interested in it. I want to see what's going on. So you click the single variant tab and voila, it takes you to this lovely page where you can input a variant to see the annotation. You don't have to run an entire job. You can use the information up here to put in your chromosome position, reference alternative allele, etc., and it will populate everything with the allele frequency, variant information, and we have some lovely examples that we recently, these are fairly recent, so these are great examples if you wanted to take a look at this single variant page. If you're in one of those research spaces where you are just doing, individual variant analysis. So we're going to move on to the part of the analysis workflow where we get to upload our file. So hopefully everyone has their SQLite file downloaded from the discussion board. If not, it's a-okay. You can follow along. This information will be just as impactful if you're following along. So to upload your SQLite files, you should be able to go to the Jobs tab, and there should be a button. My button on my particular screenshot is all the way at the bottom because I have lots of jobs that are in. But you can click Import Job, and it will automatically pop up to your file manager for your computer. Or if you're on the web browser, you can also go to the Jobs tab and click Import Job, and you'll get a box that pops up for you to upload your file. Please keep in mind that I am using version 2.7.3. So that is the reason that I see this really nice import job button. So if for some reason you have Open Cravat downloaded and you didn't get the latest version, for some reason, you should be able to click this little gear at the very top right, and there'll be an import job button, and you can get the same result. While everyone is uploading their files, I'm going to give some information on the samples that we're looking at today. So we're going to look at metastasis tissue samples. So if you guys remember previously in the beginning of the talk, we talked about the evolution of cancer in cells. So once we reach the point of metastasis, that means that the mutation has spread to other locations within the body. The samples that we have are metastasis tissue samples from warm autopsies. The data was collected from tissue samples from someone who agreed to contribute their bodies to science and medicine, and the tumors were harvested after they died. So we'd like to take a moment just to thank those persons because that's very heroic, and it's very necessary for the advancement of cancer, genomics, and science. So we have two patients here. We have our samples are coming from two people, A32 and A38. A32 is an 81-year-old male who had stage 3 pancreatic cancer. He went through 2 cycles of chemo, X-ray therapy, and from the point of being diagnosed with stage 3 pancreatic cancer, he survived for 3 months. We also have samples from A38, which is a 51-year-old male who has stage 4 pancreatic cancer, and he went through 9 cycles of chemo and a combination drug therapy. Ph.D. means that the disease began to still progress. So the initial therapy did not work, and he was given 1 cycle of rapamycin, which is also another targeted cancer drug. And he survived 15 months after his initial diagnosis. I also would like to note that I have 2 samples here, A32 and A38, because both of those files have been included on the GitHub discussion board if you guys would like to use both. However, in the presentation today, in the interest of time, I'm only going to go over A32. So I have the citation for the manuscript that these samples were initially mentioned in at the very bottom. Distant metastasis occurs late in the genetic evolution of pancreatic cancer. So that's kind of the whole point that we're going for. This distant metastasis typically occurs late, and we're trying to figure out what were those driver mutations that kind of push this to happen. So the image that we have here, this is our patient A32. It's also labeled in the manuscript as PA07, so that is what this difference in naming is. This is an image of what genes were in the primary carcinoma, so that initial tumor that they discovered. However, our samples are in the metastases. So it's going to be the peritoneum, which is the index lesion. And the index lesion is typically the largest lesion or tumor that they discover that has metastasized. And our samples are also going to be in the omentum. So we have 2 samples that should be in that zip file that everyone downloaded, and you should have been able to upload both of those files. So we have 2 samples in our file. Now I am going to move on to the actual analysis, and I want to also mention that this initial primary carcinoma began in the pancreas, and this metastasis moved on to different parts of the lining of the abdomen. So that's where the peritoneum and the omentum are. So once you were able to upload your files, you should have a screen that comes up with your job. If you were able to upload your file, you should be able to just press Open Result Viewer. Once you open your result viewer, you're going to get a large amount of information because once again, we're working with whole genome data, we're working with big data. So there's always a lot of information, but that's the power of Open Cravat. It's going to help you filter down and get to the nitty-gritty of the information that you are really trying to look into. But just to go over this summary page, there are lots of awesome widgets. So just starting from the top to the bottom, we have our input file, input genome, which is something to really be mindful of whenever you're uploading VCF files or any type of file into Open Cravat, is to make sure you're using the appropriate genome. It tells you the version, the annotators you use. It also gives us information on frequently mutated enhancers, the gene ontology summary, which is great. If it's ATP binding, RNA binding, you get all that great information. You get a summary of the samples. You also get a summary of the coding versus non-coding. I love non-coding. However, today for this particular talk, we're going to focus on coding, mainly because coding has more information in the literature, and there's more likely that that information has been annotated for just so for in the sense of time and simplicity, we're going to focus on coding. But it is really good to note that a majority of the information you see here is non-coding, and that is because most of the human genome is non-coding and only about 5% is actually coding. So this image makes a lot of sense when you think about it. We're gonna move on. So now I have moved on to the filtering tab. So I'm gonna show you guys how to build a query. So a query is just what it sounds like. So we have our file that we've uploaded with all this information. Now we need to query and we need to put in different filtering steps for the annotator to narrow down what's in the database to identify the variants that we think are drivers and variants that have been most studied. So the filters that I decided to use were variant annotation. So we want to make sure when the variant was annotated, it had some kind of data because we need data. So that's kind of a sanity check. We're also going to look at Cosmic, and Cosmic is a cancer database. And if it's in Cosmic, most likely this variant has been seen before as a somatic mutation in cancer, because remember, we're looking at somatic mutations, so because it's in the tumors. And we also want to look at cancer hotspots and see if a cancer type has been identified previously. And we want to look at cancer hotspot because typically if it is a mutation that is identified in this cancer hotspot database, the position in the genome is somatically mutated in cancers typically, and that indicates that it is indeed a driver of cancer developing further. So once you have your filters loaded and identified, you're going to press the button next to the total number of variants, which you should have about 21,309, refresh. And this refresh button is really great because it gives you the count of what your filter has identified out of the total. So you're going to do that, and then once you have that number updated, you're going to click the apply filter button. So click the apply filter button, and what's going to happen is this number up here is going to automatically change to whatever the refresh number was. So once you click the variant tab, you'll see that you have 4 variants because we identified 4 variants with cosmic and cancer hotspots and with our variant annotation information. So the 4 variants that have been identified are very likely to be candidate drivers in the metastases for this particular person developing. So we've shortened our list from 21,000, approximately 21,000 to 4. So if we look at our genes, we have 4 genes. A lot of these genes are commonly known to be drivers in cancer. Next, I'm going to go through some of the other things that you would look at in regards to cancer drivers to determine if what you're seeing on your end is correct. So to identify these driver mutations, I want you guys to expand this variant annotation tab. And once you expand this variant annotation tab, you're going to note that under the coding section, which there is a drop-down menu, if you did have more variants, you could filter this to yes or no. But we're going to focus on coding because we're looking for drivers and genes that are commonly identified as hotspots in the literature. So all of these have been identified as yes. We're also looking for information in this sequence ontology column. So 2 of our genes are missense variants, but 2 of them are also stop gains. That's really important to note because a stop gain would give some change in the actual gene function. So typically if you have some change in the gene functions, that's always going to be in that, not always, but 9 times out of 10 is in that coding region. Also, we want to look at the sample count. So this is important, especially if you're trying to identify driver mutations. So this column is letting us know that we uploaded 2 samples, and this column is saying, I have identified this particular mutation where we have this protein change, there's a stop gain, it's in both our samples. So in all 4 of these rows, we have driver mutations. So this is exciting, at least for us, because now we know that Open Cravat is doing what it said it would do, which is amazing. We can assume successfully that these mutations occurred early on in the development of the cancer because it's in all of our samples. And on the other end of this, if we were to see on our screens, if you guys take those files and you try to analyze them later on, you'll see that if it's only in 1 sample, most likely it occurred later on in the development of the evolution in the cancer of the cells. Next, I want to focus on another functionality of Open Cravat, which is the widgets. So the example that I showed you guys on the previous slide, there were 4 rows, which is great. We narrowed it down. It was easy to digest. However, sometimes you may have 50,000 variants or 100 variants or even 20. That's still a lot of information when you think about the amount of information that's out there about variants in general. So these widgets are great because it gives you a great summary of what's in your table that Open Cravat gives you. So if you click a row in your table, it highlights in yellow, and your widgets adjust and change as you click the different rows for the information. Also, it's worth noting that you can save these widgets. You can click the little camera and it will give you a really nice saved image, or you can pin it to kind of put a pin in it and keep your widget in place. So the example I have is from the first row that we just looked at. So we have aired 1A. So it gives us the information from our variant annotation tab. It gives us the chromosome position, reference, all the information we looked at on the first row. However, it gives us a lot more information in regards to what the sequence ontology might be. Typically, it's a stop gain, could be a process transcript though, depending on what transcript you may have. I also want to focus on the cancer hotspots annotator, since we are discussing driver mutations and we just talked about cancer hotspots a second ago. We can see that pancreas, which is highlighted here, has a 1 next to it. So what that means in pancreatic cancer, the exact same mutation in the gene is mutated and is seen however many times it's listed here in each cancer. So it's seen 1 time in pancreas, it is seen in uterus, bile. So this particular mutation, it seems like it really is indeed a hotspot. I also would love to point out the protein diagram. I love this protein diagram because it gives us a visual of what you see in this variant annotation tab. So we see that it is annotated our mutation as a stop would potentially be gained. So that stop gain is displayed here. And you can go through and look at the track that they have in Uniport, or if you have a reference from the variant that you have for pancreatic cancer, TCGA, they have lots of annotations that can be uploaded, and those would populate kind of at the bottom here. So these widgets are very amazing because it gives you a lot of information and it helps you kind of narrow down which variants you may think may be most important for your research or interesting, or it'll let you know, hey, maybe I need to do a deeper dive in this variant. The next thing that I would like to discuss with everyone is Nomad. So Nomad was one of the annotators that we used when I ran the job. Nomad is used as a population tool. If you got, some of you guys may know me, previously my research focused a lot on population genetics. So you'll see just from this screenshot, it gives you the allele frequencies. So you have global allele frequencies, African, Ashkenazi, Jewish, East Asian, etc. So in regards to the example that we're talking about though, this serves as a sanity check that, hey, this driver mutation that is a hotspot is low frequency in the germline. So also I want to note that Nomad is looking at germline frequency. We're looking at somatic, was in a tumor. So if these numbers were big, or if there were numbers here, it might be a little suspicious that this is a real somatic variant. So thankfully, the populations globally, this is very low, because that would be very terrible if this was a germline mutation that was, everywhere. So I am hoping at this point, everyone is excited and you're willing to use Open Cravat and trying to get some data, because variant annotation is a superhero cape in the world of medical research, shining a light on genetic mysteries. So, mysteries. I am an avid reader of thrillers, suspense, mysteries. So I love science because there's always a mystery. There's always a question. There's always a problem to be solved. So today I'm going to give you guys a peek into my own germline data that I got from 23andMe. So I'll go through a little bit of my ancestry composition, and I'll go through why I chose some of the variants that popped up in my 23andMe data. So let's get into it. This is what my ancestry composition looks like according to 23andMe. So I have listed my ancestry composition in 3 columns here: 50% confidence interval, 70, and 90. So the 90% confidence interval is really on the conservative side of things statistically. And I know as scientists, we love statistics, so I figured I'd put this in here just to pique your interest. Whenever you are looking at your 23andMe data, they automatically give you a 50% confidence interval. However, you can go in and you can look at it with different confidence intervals. But I think we can safely say that, give or take some specific regions, for the most part, sub-Saharan African, European, Indigenous American, and East Asian, and some traced ancestry that's kind of questionable. I also think it's worth noting that, especially when you're working with data and you're looking at population information, sometimes that information is as good as your reference and the data that you already have. So just something to keep in mind as we go through some of my own data. I would also like to note that if you decide to look at maybe your own 23andMe data or Ancestry, make sure you go to the Open Cravat store and install the 23andMe converter. So it's in the store. You can just type in 23 and it'll pop up. Or if you type in Ancestry, that converter will pop up. And you need this converter to convert the format that the company gives you, so Open Cravat can actually annotate it. So I was very interested in pharmacogenomics in regards to my 23andMe data. So when I began playing with Open Cravat and I saw there was a 23andMe converter, I said, oh wow. I said, this is powerful. I said, I need to get into this. And when I proposed this to Rachel, she said, oh, let's just see. And I said, I'm excited about this. This is amazing. So my 23andMe data actually showed that I am a rapid metabolizer for certain drugs. And I said, if they reported it, that means that, that's a real thing. So I looked it up and looked at my data. It was a thing. And I said, well, I wonder, is this a thing with other medications and things? I have a family history of cholesterol issues and, some other heart disease and things. So I said, okay, well, let's just look and see. So before I get into it, I just want to explain what pharmacogenomics is. So it's the relationship between genetic variants and how your body responds to medications, which I think is so interesting because sometimes you go to the doctor, they prescribe medications, you get these pamphlets with all these side effects, and you just never know sometimes how you're going to respond. So for my 23andMe data, I took my 23andMe data file, I uploaded it, chose all of the annotators that you guys use today, minus the cancer annotators, and I just included the form GKB annotator. So to do my filtering, because I had over 100,000 variants that were identified, I narrowed it down to 45 using variant annotation, looking at coding, form GKB, which was the pharmacogenomics annotator, and I looked at gene enhancer because I said, maybe if the variant is enhancing or affecting the actual gene expression, this will be interesting to look at. So those were the filters I chose. For the example today, I have two variants. So the first variant is RS5522. So this particular variant, I had the genotype TT, and you can tell just from the widget that we have from variant annotation. Typically, the amino acid changes from a G to A. It's a missense variant, and these are the different transcripts that you could have. So I am homozygous for TT. I said, okay, homozygous for alternate base. That's interesting. Let's see what this means. Also, gene enhancer gave me some nice target genes that I might have some linkage with the genotype I have. Good to know. This is a screenshot of my 50% confidence interval and what it looks like for this variant in other populations. So it seems like the variant that was identified has a pretty high frequency in other populations. However, I am homozygous for the TT variant. It is also good to note that if you go to the actual Nomad website and you put in the RS variant ID number, it will give you a breakdown of the variant It'll give you a breakdown of how much is seen in each one of these populations, as well as what it looks like in male versus female. So you get kind of a broad view of what's going on, but you don't necessarily see like how many people have TT or CT, etc. So just keep that in mind if you decide to look at Nomad. So I used the information that showed up on my table viewer in Open Cravat in the Form GKB tab. So I expanded my tab. We already know I'm homozygous over here with the variant metadata. So it says that I have an efficacy for enalapril. And I said, okay, well, I have no idea what enalapril is because I'm not a pharmacist. I'm not a medical professional. None of us are medical professionals here, I don't believe. However, they give you a nice little ID number that's hyperlinked that you can click. So I said, okay, let's click it. I click it and I get into it. So before we move on, I want to note that efficacy is the ability of an intervention, so a drug or a surgery, to produce the desired beneficial effect. So I go down, I look at the descriptions in my widget, and you can click this hyperlink to get more information. But once you go to the widget, you can see that my homozygous genotype is associated with an increased response to enalapril in people with hypertension. And I said, well, that's good to know. So if I develop hypertension later on, this is information that's beneficial to me and my health. It's also good to note that whenever you click the hyperlink, or even if you look at the widget, it lets you know what category that they have put this variant in, if it's the information from the research study was significant or not. They give you a link to hyperlink to the actual study, and it gives you some really handy dandy notes and summary about what was actually in the study. I will say that not that some of the variants, if you go through and you look at them, they may have more than one study. Some of them may have one study. I think you kind of have to also realize that in regards to personalized medicine and looking at variants, sometimes different populations are studied more than others, or maybe the information just is not there for a certain population. So it may be that the information was there for a European population, but not for African American or Asian. So I found that very interesting, especially for me, considering the admixture that I have in my ancestry. The next variant that I'm going to show you guys is a variant that is associated with cholesterol. And my genotype was homozygous GG. So specifically, I was really, I won't say excited, but I was interested in learning more about the pharmacogenomics of medications with cholesterol. So one of my very close family members, she had cholesterol issues. She was taking medications, statins, and she ended up having a allergic reaction to it, muscles degrading, and she eventually ended up passing just from all the complications. And when we were in the hospital, the rheumatologist said, hey, this could be something that's genetic because it progressed so quickly. So just be mindful that maybe, be aware of what you're taking and be mindful. So especially with this particular drug I'm going to show you, I thought it was very interesting. So we look at the nomad population, and we can see that, in some populations, African American, East Asian, South Asian, it seems to be a little more predominant. We saw from my 23andMe that I have some African and Asian mixed in. So, okay. It seems like from this other annotated graphs that there's significance of this variant being associated with GWAS studies and other phenotypes with cholesterol, hip waist ratio. So I said, okay, well, interesting. So I move into the pharmacogenomics. And we have the culprit, atorvastatin. So statins were the medication that my family member was having an adverse reaction to, actually. So this is interesting to me in particular because I'm homozygous for this particular variant. And this study was saying that the genotype homozygous AA is associated with an increased response. However, if you have the other variants, heterozygous or homozygous for GG, you do not have you don't have a increased response as you would if you had the other genotype. It was significant, and they gave me some little summary notes, and I had a couple variants that were actually associated with sadness, and I said, well, this is something for me to really keep in mind. So I'm hoping that this 23andMe example impresses the importance of using open source software and software in general, like OpenCRAVAT, not just to annotate your own data, but the importance of it and the impact that it has on personalized medicine, genomics, and big data, and being able to make a positive influence and impression on science for generations to come. I hope that everyone has enjoyed the presentation today. I hope you were able to follow along. If you have any questions, please let me know. The chat box is open. I want to thank everyone for exploring Open Cravat's powerful features. I want to thank Doctors Christy and James for sharing the data with us that we used today for this tutorial. I want to thank the courageous donors for believing in the advancement of cancer research. And thank you to the Open Cravat team, who I believe you guys are here today. Thank you, thank you, thank you. And the Black Women in Computational Biology Network. We also have our funding source here. We'd like to thank NCI. And if you guys would like to cite us, there's the information that you can use. Please connect with us if you're on social media. This is our Twitter, Big Sky, and Mastodon handles. I also have QR codes here for our GitHub Open Cravat page. If you are on GitHub and you're willing to contribute to our annotator or even just say hello, pop into our discussion board. We also have the QR code for our discussion board here. And thank you, thank you. I have the link to the feedback survey here. So if everyone wouldn't mind, you could please scan the QR code to get the feedback survey. I am eager to read the feedback and incorporate it and see what you guys thought about Open Cravat, and how you might like to use it for your research. I am going to paste the link. So the link is in the chat box for the feedback survey. Please take a few minutes to fill that out. It shouldn't take more than 2, honestly. And we have 5 minutes left, so we're gonna, if anyone has any questions that hadn't been addressed in the chat box, please feel free to unmute yourself and let's give it a go. I hope everyone enjoyed the presentation.
Yeah, that was great. Thanks so much, Jasmine. There's one question that I saw from Sarah Cooper. Did you want to unmute and ask it yourself, or I can read it? The question is there a maximum file size for VCFs as input, or is that just based on whatever your system can handle?
So once you go to the setting, the little gear in the top right, you can adjust the input size.
And then we have another question from Deanna. I'm hoping I'm saying your name right. She asked, does Open Cravat take Plink input files?
Currently, Open Cravat does not take Plink input files. And hello, Deanna, I know where you're going with this. So we can talk offline about that in more detail. I'm assuming that you may be wanting to use it for some of your research that you're potentially using with the All of Us program, maybe, or some other extension of a large database. So if that's the case, just pin me a message and we can discuss further. But currently, as of right now, we do not take link files as input files.
Alrighty, we are 1 minute over time. I'm just gonna quickly go through some of our announcements here. For those that don't know, the Black Women in Comp Bio, we have a podcast called the Comp Bio Cafe. We released a new episode with the title Thoughts on Conference Diversity, Belonging, and Accessibility. So please feel free to check that out. We were recently in Boston at Festival of Genomics and Biodata, and we had an amazing time both at the conference and at our collaborative first time in-person event where we collaborated with the Boston area women in bioinformatics to put on this fireside chat. We had lots of help from our partners also to help put on this event, and we had a great time. With these ladies kind of just sharing their knowledge on going through different career hurdles. We'll be at ISMB in next month. So if you're there, definitely come by. We'll have a table. We'd love to meet you guys in person. And then also want to shout out our grants that we're able to give out this year. We gave 2 travel grants and then 1 Accelerate Growth grant. And yeah, that is it. I don't know if we have been able to share our links in the chat here. But yeah, we have a merch site if you want to go in and shop some merch. But yeah, thank you all for coming. Any other final words? Are we good? All right. Thank you all for coming. Have a great day. Don't forget to give us some feedback and yeah, keep engaging with us. Thanks, Jasmine.